the microbiome, and the central nervous system. The human microbiota is the collection of microorganisms that exist on and inside each human being, and trillions of microbes are present in the gut lumen. The use of high-throughput gene sequencing technology has allowed for culture-independent testing of the microbiota to determine the microbiome, the collection of microbiota genes. Using these advanced techniques, it is possible to simultaneously analyze all microbes in the gut. Information gleaned from this research has fundamentally changed our understanding of the brain-body interface. Starting at the gut, we'll briefly examine how the microbiota influences the body and the brain, and we'll examine some of the possible clinical implications of these connections. Under normal circumstances, a balance exists between gut microbes and the gut epithelia. Gut microbiota are essential for digestion, nutrient uptake, and vitamin synthesis. Changes to the composition of the gut microbiome result in gut dysbiosis, and gut dysbiosis has been correlated with diseases, especially those related to inflammation, such as ulcerative colitis or Crohn's disease. An array of mechanisms are in place to ensure symbiosis and separation between the microbiota and the gastrointestinal epithelia. Mucus is produced by goblet cells and arranged in layers so that the outer layer, more distant from the epithelial cells, has a higher microbial concentration than the inner layer of mucus. Additionally, gut epithelia secrete antibacterial proteins either continuously or in reaction to a stimulus. For example, toll-like receptors are expressed on gut epithelia, and when pathogen-associated molecules bind these receptors, they can synthesize and release antimicrobial proteins into the mucus. Dendritic cells located in the gut-associated lymphoid tissues are able to take up bacteria and then stimulate lymphocytes to produce immunoglobulin A. Bacteria that penetrate these defenses are ultimately phagocytized by macrophages associated with the gut epithelia. A key component to the symbiosis between the gut and the microbiota centers around the substrates produced by the bacteria. Commensal bacteria tend to produce small-chain fatty acids, which are essential for host physiology. A commonly seen small-chain fatty acid is butyrate, which is used as an energy source by the gut epithelia to maintain health and integrity of the epithelial barrier. Selective loss of butyrate-producing bacteria can lead to dysbiosis, and treatments that restore butyrate production can overcome the effect of gut dysbiosis. While the existence of pathways between the gut and the brain, termed the gut-brain axis, is still a relatively new concept in biology and medicine, multiple reports have revealed the important role that the vagus nerve plays in coordinating gut-to-brain and brain-to-gut communication. The vagus nerve innervates the gastrointestinal mucosal wall at 3 sites: the gastric antral glands, the apices of the villi, and at the crypt-villus junction. These nerve endings appear to be located below the epithelium, so interaction with luminal bacteria is normally indirect and through diffusion of metabolites such as butyrate, or through interactions between the vagal afferents and enteroendocrine cells. The vagal afferents from the gut serve as the sensory link to the brain and can signal inflammation or inhospitable conditions. Vagal receptors in the gut are varied and include mechanoreceptors, osmoreceptors, and chemoreceptors, and these vagal afferents mainly terminate at the nucleus tractus solitarius. To summarize, projections from gut vagal afferents project to brainstem nuclei, such as the nucleus tractus solitarius, which then project to hypothalamic nuclei, such as the arcuate nucleus, for signaling satiety. In the normal state, vagal chemoreceptors bind ligands such as butyrate produced by probiotic commensal bacteria. However, afferents can also be activated by pathobians. For example, in animal models, inoculation with Campylobacter jejuni has been shown to increase neuronal activity both in the gut and in the nucleus tractus solitarius within 4 hours of inoculation. This pathologic activation of the nucleus tractus solitarius by viscerosensory afferents correlated with decreased exploration and increased anxiety, suggesting that gut microbe-derived signaling affects behavior via vagal sensory afferents. Given the numerous projections from the nucleus tractus solitarius, there are multiple potential pathways by which gut microbes may potentially influence central nervous system function in both normal and disease states. In preclinical models of traumatic brain injury and spinal cord injury, significant alterations in the composition of the microbiome develop shortly after injury and are characterized by a decrease in lactobacillus and an increase in Clostridial genera. Furthermore, pre-injury disruption of the microbiome may exacerbate neurological injury. The following figure demonstrates a mouse model of spinal cord injury, where dysbiosis was induced prior to injury by treatment with a cocktail of antibiotics. Antibiotics were administered from 7 days before injury to 14 days after injury. Following spinal cord injury, injured animals with dysbiosis were compared with similarly injured animals that had not been given antibiotics. Injured animals with induced dysbiosis were noted to have worsened locomotor recovery, larger spinal cord lesion volume following injury, and increased spinal cord infiltration of macrophages, B cells, and T cells. This is just one example of how the composition of the gut microbiota may influence outcome following a neurological insult. With the advent and commercial availability of genetic tools for studying microbes, researchers have recently uncovered both the diversity and importance of the gut microbiota. Future research will further advance our understanding of how altering the gut microbiota, either naturally or through direct intervention, may impact human health in both the normal and diseased states. As we continue to uncover the cellular, molecular, and anatomical mechanisms of the gut-brain axis, we will likely further understand how to manipulate these pathways both to improve health and to treat illness.